Hi, my name is Leo Lebanov and I'm going to present to you preclinical studies of Invirase with its active compound saquinavir. Invirase is an antiviral medication that prevents human immunodeficiency virus cells from multiplying in human body. Invirase with its active compound saquinavir is highly selective inhibitor of human immunodeficiency virus enzyme, HIV protease. Infects a host, it directs the synthesis of several polyproteins. The maturation of virus to its infectious form requires that these polyproteins be cleaved to their component proteins by HIV protease. The subunits of HIV protease form together catalytic tunnel capable of binding the nascent peptides and cleaving them into their mature form. Within this tunnel are two aspartic acid threonine glycine conserved sequences, which contain the catalytic aspartic residues. These catalytic aspartic residues carry out the hydrolytic cleavage of the polyproteins. Saquinavir binds very precisely to these conserved sequences within the HIV protease tunnel, preventing the nascent polyproteins from entering. Unable to actively cleave the nascent proteins into the appropriate form, HIV is unable to mature and proliferate, allowing the patient's immune system to fight off the infection more easily. In vitro antiviral activity of saquinavir was assessed in lymphoblastoid and monocytic cell lines and in peripheral blood lymphocytes. Saquinavir inhibits HIV activity in both acutely and chronically infected cells. IC50 and IC90 values were in the range of 1 to 90 nanomoles and 5 to 80 nanomoles respectively. The concentration of saquinavir needed to inhibit by 50% activity of human aspartic protease are 10 micromoles or even more, compared with typical IC50 of HIV protease, which was in range of 1 to 10 nanomoles. The pharmacokinetic profile of saquinavir was determined in several species using radio-labeled saquinavir material and adequate methods. The absorption after administration of a single oral dose of radio-labeled saquinavir in bio-duct cannulated rats was low, about 15%, with corresponding low oral bioavailability, 2.5%. In all species studied, the binding of saquinavir to plasma proteins was more than 98% and appeared to be independent of concentration. Tissue distribution was investigated in rats after a single oral dose of radio-labeled saquinavir equivalent to 10 mg per kilogram and an intravenous dose equivalent to 1 mg per kilogram. Organ plasma ratio after intravenous administration was about 6 to 8 and strong binding to melanin was evident. Saquinavir crosses the brain-blood barrier to a limited extent and levels in the central nervous system was found less than 10% of plasma levels. These results suggest that saquinavir may be less active against viruses localized in the brain. The potential passage of the placenta barrier was very low. Metabolites characterized in all species consisted mainly of mono and dehydroxylated derivatives, and it was demonstrated that CYP3A4 was the major isoenzyme responsible for the biotransformation of saquinavir. After parenteral administration in laboratory animals, it was shown that the main elimination pathway was via the bile and feces, more than 95%, and in rats, the administrated dose was almost totally recovered within 3 days. The renal elimination of saquinavir was negligible. Acute oral toxicity studies were performed in mice, rats, and marmosets, and results from these studies showed that saquinavir was well tolerable, and this picture depicts it. Repeated dose toxicity was studied in mice, rats, dogs, and marmosets up to 1 year. In spite of plasma exposure approximately 7 to 40 times higher than clinical exposure, the toxicity profile of saquinavir in animals could not be clearly and consistently identified. The toxicological findings in oral studies were mild and often transient. reversible, inconsistent in nature, and were in general not accompanied by histological changes. Functional evidence of hepatotoxicity increases in serum aminotransferases, alkaline phosphatases, and isocytic dehydrogenases, was noted in rats and in high-dose groups of marmosets and dogs. A potential hepatotoxicity of saquinavir cannot be excluded at high exposure levels. A special study was carried out in rats aged between 4 days and 9 weeks. After oral administration of saquinavir, an increased susceptibility and increased mortality, probably due to gastrointestinal changes, was observed. Results obtained from a complete program of reproductive studies indicated that saquinavir was not associated with any reproductive, teratogenic, or developmental effects in rats and rabbits. Systemic exposure in these studies was 1.5 to 5 times higher than clinical exposure. The actual frequency of congenital defects possibly related to saquinavir therapy is currently still unknown. Carcinogenicity studies performed in rodents did not evidence any carcinogenic potential of saquinavir. Invirase has two dosage forms: 200 mg hard capsules and 500 mg film-coated tablets, which contains 571.5 mg of saquinavir mesylate, equivalent to 500 mg of saquinavir free base. 200 mg hard capsules contains 200 mg of saquinavir, the saquinavir mesylate. Each capsule or tablet contains of many inactive ingredients. Studies have showed that the best package for 200 mg hard capsules is 270 tablets, and for 500 mg film-coated tablets, 220 tablets per package. Invirase capsules and tablets are administered orally. For infants and children unable to swallow the Invirase capsules, mix the content of Invirase capsules in sugar syrup and sorbitol syrup. For children with type 1 diabetes or glucose intolerance, jam or baby formula.